In today's topic, we will study the definition of micturition, anatomy of the bladder. Under anatomy, we shall study the detrusor muscle, internal and the external urethral sphincter, then the nerves of the bladder, which includes the sympathetic nerve, parasympathetic nerve, pudendal nerve, and from where they arise, Laplace's law, control of micturition, and abnormal bladder that includes autonomic, atonic, and neurogenic bladder. Micturition. The urine formed by the kidneys drains via the two ureters and collects in the bladder where it is temporarily stored. When the bladder becomes full and the surroundings are favorable for voiding, the subject passes urine. The process of voiding urine is known as micturition. Anatomy of the urinary bladder. The bladder wall is composed of a muscle called the detrusor. It consists of a transverse, longitudinal, as well as spiral muscle fibers. Contraction of detrusor raises the intravesical pressure and leads to voiding of urine. Intravesical pressure is the pressure within the urinary bladder. Around the neck of the bladder, there are muscle bundles forming the internal urethral sphincter. This sphincter remains contracted except at the time of voiding urine when it relaxes. The muscles of the internal sphincter are so disposed that whenever the bladder contracts, the internal urethral sphincter is pulled open. There is another muscle located more superficially, that is, the external urethral sphincter. This encircles the urethra and is under voluntary control. It prevents reflex passage of urine and keeps voiding of urine under voluntary control. Urine enters into the bladder from the two ureters aided by gravity as well as by peristaltic contraction of the ureters. The ureters pass obliquely through the wall of the bladder. As a result, they are compressed at this region when the bladder fills. This prevents backflow of urine into the ureter from the urinary bladder. The bladder acts as a temporary reservoir of urine. The epithelium and smooth muscles of the bladder are specially adapted for this reservoir function. As the bladder fills, its folded walls flatten out to increase its volume. At the same time, the transitional epithelium of the bladder increases in surface area. The vesicles in the epithelial cell are extruded by exocytosis. This adds membrane to the cells and the epithelium expands. On the other hand, when the bladder empties, vesicles are endocytosed, causing shrinkage of cells and epithelium. The smooth muscles, because of their special property of plasticity, allows the urine-filled bladder to be distended considerably without any appreciable rise in intravesical pressure. Nerves of the urinary bladder. The bladder has three different sources of nerve supply. The first one is the sympathetic nerves. These arise from the intermediolateral gray column of the spinal cord from L1 to L3 spinal segments and traverse along the hypogastric nerve to the urinary bladder. The postganglionic fibers supply the detrusor and the internal sphincter. On stimulation, they cause relaxation of the detrusor and contraction of the internal sphincter. Hence, they are called nerves of filling. However, this role of sympathetic nerve is somewhat controversial. It is now thought that the sympathetic nerves are chiefly involved in preventing reflux of semen into the bladder during ejaculation. The second one is the parasympathetic nerves. They arise from the sacral outflow of the parasympathetic system. Arising from S2 to S4 spinal segments, they pass along the pelvic splanchnic nerves. The postganglionic fibers supply the detrusor and the internal sphincter muscles. They cause contraction of the detrusor and relaxation of the internal sphincter. So, they are called nerves of voiding. The third one is the pudendal nerve. This is a somatic nerve which supplies the external urethral sphincter. So, points to remember: sympathetic nerves arise from L1 to L3, supply the detrusor and the internal sphincter, causes relaxation of these muscles, therefore, they are called nerves of feeling. Then we have the parasympathetic nerves, arise from S2 to S4, supplies the detrusor and the internal sphincter, causes contraction of the detrusor and relaxation of the internal sphincter, called the nerves of voiding. And then we have the pudendal nerve, which supplies the external urethral sphincter. This is the diagram. This is the urinary bladder, sympathetic nerves which arise from L1 to L3, parasympathetic nerves which arise from S2 to S4, and the pudendal nerve. Sympathetic nerves supply the internal urethral sphincter and the detrusor muscle. Parasympathetic nerves also supply the internal urethral sphincter and the detrusor muscle. Pudendal nerve supplies the external urethral sphincter. Point to remember: the urinary bladder follows something called as the Laplace's law. So the bladder, which is almost a thin-walled spherical organ, obeys Laplace's law. According to this law, the transvesical pressure, that is the pressure difference between the inside and the outside bladder, is equal to twice the wall tension divided by the radius of the bladder. So P, which is pressure, is equal to twice of T, that is twice the wall tension, divided by R, that is radius of the bladder. When the bladder is distended by urine, wall tension rises, that is T rises, but due to relaxation of detrusor owing to plasticity, there is increase in diameter of the bladder. As a result, the transvesical pressure does not rise and is maintained at the same level, that is 2T divided by R remains the same. Control of micturition. When the volume of urine in the bladder is 150 ml, sensation of bladder fullness is felt. When bladder volume is 400 ml, there is a strong urge for micturition. At 600 ml, pain is felt. Urination may be delayed by voluntary contraction of external urethral sphincter and perineal muscles until a suitable surrounding is found. When the bladder is full, the stretch receptors in the bladder wall are stimulated and they tend to reflexly inhibit or relax the external sphincter so that urination may occur. But if the surrounding is not suitable, this is overridden by descending excitatory input from cerebral cortex to the motor neurons supplying the sphincter. As a result, the external sphincter closes tightly and urination is prevented. Micturition can be initiated voluntarily even when the bladder is not full. Here, the pelvic floor and external urethral sphincter are relaxed voluntarily. The lowering of pelvic floor stretches the bladder wall and opens the internal urethral sphincter. This activates the micturition reflex. Concomitant contraction of abdominal muscles and the diaphragm by raising the intra-abdominal pressure helps in evacuation of the bladder. During micturition, The external urethral sphincter and the perineal muscles relax, while the detrusor contracts. The last few drops of urine in the urethra in the male are expelled by contraction of bulbocavernous muscle. In contrast, gravity aids the emptying of the female urethra. Although voiding of urine is to some extent under voluntary control, it is essentially under supraspinal control. When the bladder distension reaches a critical level, visceral afferent fibers are stimulated. They ascend along the spinal cord to stimulate Barrington's center in the rostral pons. Abnormal conditions of bladder. The first one is autonomic bladder, when there is damage to the spinal cord above the spinal centers of micturition at the sacral region. Micturition reflex may remain intact, but voluntary control over micturition is lost. Soon after the injury, micturition reflex is temporarily lost due to spinal shock. However, this reflex reappears if the shock stage is well managed, as the entire pathway of this reflex is intact. Reflex micturition occurs periodically when the bladder is distended by urine. However, there is no voluntary control. Some patients learn to utilize mass reflex, in which urination, defecation, and autonomic changes occur on scratching the inner aspect of the thigh in order to gain some control over toilet functions. The second type is atonic bladder. In this type of abnormality, the afferent pathway of micturition reflex is damaged. This is typically seen in tabes dorsalis, a lesion found in neurosyphilis involving the dorsal root. Here, although the bladder is distended with urine, micturition reflex is not activated as the damaged sensory pathway fails to sense the stretch. As a result, the bladder is distended, thin-walled, and hypotonic. Urine overflows from the overfilled bladder in dribbles. This is known as overflow incontinence. The third type is excitable neurogenic bladder. In this, lesions in the spinal cord involve only the higher inhibitory pathways which control micturition reflex. As a result, the bladder becomes hyperexcitable. Micturition is frequent due to the hyperactive micturition reflex, which is activated even when the bladder contains small volumes of urine.